Flyworm and Weed Part 1. The International Human Genome Project. The International Human Genome Project, initiated by the U.S. National Institutes of Health and the U.S. Department of Energy, was launched in 1990 with completion due in 2005. However, in the meantime, a private enterprise, Celera Genomics, embarked on a project with the same aim but a different approach and actually overtook the Human Genome Project. The competition was settled with a historic press conference on 26 June 2000 when US President Bill Clinton, J. Craig Venter of Kerala, or um, or Celera Genomics, and Francis Collins of the National Institutes of Health jointly announced that a working draft of the human genome had been completed. A working draft of the human genome had been completed. Okay, next we have many commentators have qualified this announcement as more a matter of public communication than a scientific achievement. At the time, the accepted criteria for completion of a genome sequence, namely that only a few gaps of known size remained to be sequenced and that the error rate was below 1 in 10,000 base pairs had not been met by far. The euchromatin part of the genome was not completed until mid 2004, although that milestone was again considered by some to be only the end of the beginning, in 2004. Nevertheless, the Human Genome Project can be regarded as one of the most successful scientific endeavors in history, and the assembly of the 3.12 billion base pairs of DNA requiring some 500 million trillion sequence comparisons was the most extensive computation that has ever been undertaken in biology. The number of organisms whose genomes has been sequenced completely and published is now approaching 300. Bacteria dominate the list as the small size of their genomes makes these organisms well studied for whole genome sequences. By June 2005, no fewer than 730 prokaryotic organisms and 496 eukaryotes were the subject of ongoing genome sequencing projects. List of complete and published genomes. Bacteria total 211, many common laboratory models and pathogens. Archaea total 21, and several methanogens and extremophiles. Eukarya myxomycota 1, dictyostelium, discoideum, slime mold. Entamoeba 1, 4 plasmodium, Entamoeba histolytica, amoeba causing dysentery. Epicomplexa 6, 4 plasmodium and 2 micro my eukarya we have myxomycota 1, dictyostelium, discoideum, slime mold. Entamoeba 1 for Entamoeba histolytica amoeba causing dysentery. Apticomplexa 6, 4 Plasmodium and 2 Microsporidium species. Kinetoplastida 2, Trypanosoma brucei, Leishmania tropica parasites. Cryptomonadina 1, Gilardia theta flagellated unicellular alga. Bacillariophyta 1, Thalassiosira pseudonana marine diatom. Rhodophyta 1, Cyanidioschizon Meroli small unicellular red alga. Plants 4 Chlamydomonas Reinhardti green alga Populus Trichocarpa black cottonwood Arabidopsis thaliana Thale cress Oryza sativa var Japonica var indica rice, including fungi 14 including Saccharomyces cerevisiae, baker's yeast. Animals nematoda 2. Animals nematoda 2, Caenorhabditis elegans free living roundworm, Caenorhabditis briggsii, Bombyx mori silkworm, Drosophila melanogaster fruit fly, and Anopheles gambiae mosquito malaria vector, Apis mellifera honeybee, Tunicata 1, Siona intestinalis sea squirt, Pisces 3, Tachyfugu rubrips, puffer or fugu fish, Tetrodon nigroviridis, pufferfish, Danio Rio zebrafish. Aves 1, Gallus red jungle fowl. Mammalia 5, Rattus norvegicus brown rat, Mus musculus house mouse, Canis familiaris domestic dog, Pantroglodytes chimpanzee. Homo sapiens human. Animals total 16, eukarya total 47, overall total 279.